Hello. Thank you very much, Professor Bonora. Thank you very much to the organizing team for giving me the opportunity to speak to you today and to present my research on circulating microRNAs and diabetes risk. So I was asked to, as an entry slide, to briefly give the audience an overview of what I'm doing and to describe my team. So I'm leading the clinical epidemiology team in Innsbruck, which is an interdisciplinary team of epidemiologists, mathematicians, and clinicians, and we work a lot in international collaborations. So the aim of our group is to apply cutting-edge epidemiological methods to really, based on large-scale epidemiological data, to answer big questions with big statistical power. Of course, there's a primary focus in my research on cardiovascular disease, but also on metabolic diseases, and we try to go beyond that as well. And here just some examples. So the top on the right is my work on that I did back then during my PhD, looking at prediction models for cardiovascular disease and the added value of natriuretic peptides in predicting risk for coronary heart disease, stroke, and heart failure. But we're also doing a lot of research into evaluating the etiological role of different risk markers, including lipoprotein A, for example, or this is an overview of studies that we have been collating on subclinical markers of atherosclerosis, where we look at the predictive value of, for example, intima media thickness or carotid plaque for future cardiovascular risk. So these are my conflicts of interest. I have to declare that I'm a named inventor on a patent related to microRNA 122 in metabolic disease. So let me start by introducing to you what microRNAs are. So microRNAs are long, so they're small non-coding RNAs. They have a length of about 22 nucleotides. And what they do is they regulate gene expression at a post-transcriptional level by binding to the 3' untranslated region of their target messenger RNAs. By binding to them, they then induce direct degradation, or they repress the translation of this messenger RNA. So what is quite fascinating is that these microRNAs, they form a very complex regulatory web in different processes in the human body. So what they can do is that they, as you can see here, they can affect and target several targets in the same biological pathways. They often also closely interact with each other, and what we also see, which seems quite counterintuitive initially, but they may also both interact with activators as well as inhibitors of a functional regulator in a specific biological process. And by doing so, they kind of constitute a layer of epigenetic regulation of different processes in the human body that are particularly sensitive, for example, cell growth, differentiation, stress response, or tissue remodeling. And they thereby are thought to safeguard the stability of biological systems in the human body. So microRNAs, they act at the cellular level, but they can also be detected in circulation, which is obviously quite important for us because we can then study them in large-scale epidemiological studies. So the fact that they can be found in circulation was first shown about 10 years ago by a group in Seattle, which measured 3 different microRNAs using quantitative PCR. and they observed a remarkable stability. So we've written a review back in 2012 about microRNA compartmentalization, and one thing that we do know about microRNAs is that they often occur in different microvesicles, for example, microparticles or exosomes, so they are packaged into those, and this protects them from degradation by different enzymes in the blood. Those microvesicles in the blood are released by various different cell types in the human body in different circumstances. So there's speculation that when someone has a certain disease, it might be diabetes or myocardial infarction, etc., that specific cells release specific microRNAs in circulation, and those can then serve as a readout to the researcher or the clinician in the blood. Good. So we can use those and we can measure those in circulation. So for the talk that and the different data that I will be presenting to you today, I will largely focus on a specific microRNA called microRNA 122. This is a microRNA that is liver-specific. It actually constitutes about 70% of all the microRNA that is produced by the liver. We know that microRNA 122 may play some role in fatty acid oxidation and lipid synthesis, and there's actually also an antisense oligonucleotide that is being tested or has been tested in a phase 2 randomized controlled trial in patients with hepatitis C, called miravirsin. So this was published a couple of years ago in the New England Journal of Medicine. And you can see that with that drug, the viral load of patients that received the intervention was reduced, potentially because it interferes with the assembly of the hepatitis C virus. And most importantly, this is also a microRNA that is detectable in circulation. There are several things, unfortunately, or back then, that was unknown. So we didn't know the precise mechanisms and of how microRNA 122 reacts or interferes with the lipid metabolism. And most notably, there are just very, very few. There were very few data out there in human populations, some case control studies and quite small studies, and we were really interested in quantifying the long-term risk of people that have high microRNA 122 and their risk of metabolic and cardiovascular events. So this is something that has motivated us to do the project I present to you today. So to address this uncertainty, we conducted a set or several different analyses using different kind of data. We used human data as well as mechanistic data from different mice and cell culture studies. We were interested in the correlations or correlates of microRNA 122. Is a high level associated with higher age? Is it associated with being obese, etc.? But we also went into a layer furthermore into detail by using data on lipidomics and proteomics in our studies. We then also looked at what effects a statin intervention might have on microRNA 122, and then of course the final and most important step then is to look at the association with incident disease over a follow-up time of 15 years. And we combined the human data, we supplemented that with experimental data on microRNA 122 inhibition in mice and the statin intervention in cell cultures. So now let's start off with the epidemiological parts, and most components there are based on epidemiological data from the Bruneck study, which is located in the northern part of Italy, in South Tyrol, in the city of Bruneck, or Brunico in Italian. So this was a study that was initiated already a long time ago, back in 1990, where almost 1,000 individuals were recruited. They underwent a really comprehensive testing back then, including an oral glucose tolerance test. And those same study participants were then followed up every 5 years since. So we actually conducted an additional investigation in 2016 as well, but this it wasn't part of that particular analysis. What we then did is that we measured microRNA 122 at 2 time points in 1995 and the year 2000. We measured that in duplicates. We measured it in serum and plasma samples, and this resulted then in measurements of over 3,000 samples using a quantitative PCR method. And then our aim was to relate the level people had back then in 1995 to the risk of clinical events over time, where we could use a 15-year time frame, so events that occurred up to the year 2010. So now let's focus about the correlation and correlates of microRNA 122. And first of all, this is just the first step in that analysis. We looked at the correlation and agreement of levels in using different sample types. So you can see that there was a remarkably high correlation of the values in plasma and serum samples with a partial correlation coefficient of 0.86. On the other hand, what you can see on the right side is when we looked at the variability and stability of levels over time, it was actually quite poor. So there was only a very weak correlation between the values we measured in 1995 and the values we measured 5 years later. So there seemed to be some changes in the individuals over time. Now, let's have a look at major clinical characteristics in these study participants. And you can see here the different categories that we decided to look at. And I just want to walk you through quickly. So you can see here the direction of the association that we observed. And this is the magnitude of association. So these are percentage changes in the microRNA 122 per standard deviation higher level of these characteristics or compared to a reference group. And in summary, so what we saw in general, that microRNA 122 was correlated with an adverse metabolic profile. It included a very strong positive correlation with the liver enzymes, which was of course expected. It was also higher in people with higher levels of adiposity. It was positively correlated with inflammation, with insulin resistance, but also with an adverse lipid profile. So positive correlations with total and LDL cholesterol and triglycerides, and an inverse correlation with HDL cholesterol. And strikingly, the most important or numerically biggest, strongest association was observed with pre-existing diseases. So people with diabetes or with a metabolic syndrome had markedly higher levels of microRNA 122. So now let's go one step further, and we looked at the correlation with lipidomics measurements. So this is some data that we already had available from a previous study, where we looked at cardiovascular disease, which was published in Circulation. So using shotgun lipidomics with mass spectrometry, we were able in this study to quantify a total of 135 different lipid species, which you could allocate to 8 different classes. And these data are it's very difficult to kind of like think about how to present these kind of data effectively, because the data, you have so many different lipids, right? But we kind of thought we'll do it that way. You can see here, these are the 8 different classes. and then you can differentiate it or distinguish between lipid species according to the number of single bonds that you can find in them and the number of double bonds. And the individual bubbles here, so the color indicates the direction and the magnitude of correlation, and the size of the bubble indicates the p-value of the correlation with microRNA 122. And one thing that we could identify there, and this is really a key finding, is that there's a preferential association with lipid species that contain saturated fatty acids as well as monounsaturated fatty acids, and those are some that can be produced by the liver by de novo lipogenesis. All right, so now let's look at proteomics. So we also quantified 93 different proteins in this study population using liquid chromatography mass spectrometry. And I just want to show you here the most important results. So first focusing on the different types of apolipoproteins that were measured using this technique. First of all, here marked in blue, you can see an inverse association with ApoA4 and ApoD associated or with HDL and chylomicron particles, but also we found positive associations with a range of different apolipoproteins associated with the VLDL particle. Among the other proteins that we looked at, the top hits were, first of all, affamine. So this was actually the strongest positive correlation. And we know that affamine is associated with the metabolic syndrome in several different population-based studies. And affamine also, similarly to microRNA 122, can be found in exosomes produced by the liver. Then we also found a positive association with the complement factor H associated with oxidative stress, and zinc alpha-2 glycoprotein, and that's a protein that is known to be important for insulin sensitivity. So these kind of correlations in terms of the biological function made sense and were plausible. Okay, so as a next step, we then ask the question: what happens when you then inhibit microRNA 122 in mice? And so there we used a specific inhibitor called the antagomir 122. And this was successful, resulted in complete inhibition of microRNA 122 in those mice, and as expected, we also found a reduction in total cholesterol. When we then looked at the gene expression and the effects on gene expression, we saw a downregulation of 3 different genes, so associated with fatty acid synthesis, VLDL assembly, as well as glucose metabolism. At the protein level, we also saw some differences, so those are so-called volcano plots, where you can see the fold difference, fold changes upon intervention and the p-value, and you get the most prominent changes up here. And so some of them include also different proteins, again associated with fatty acid metabolism, hepatic HDL release, and VLDL removal, and then also another protein where we know there's a deficient mouse model that has been studied, where we also know that this mouse has reduced total cholesterol levels. Okay, so what happens now if in such a system we then intervene with statins? What does it do with microRNA 122? And we were able to, through a collaboration with London, to use data from the so-called ASCOT trial, which is a double-blind, randomized 2x2 factorial clinical trial that evaluated on one hand lipid-lowering therapy, on the other hand blood pressure-lowering therapy. For the lipid-lowering component, people were randomized to receive either atorvastatin 10 milligrams per day or placebo. And what we did and what we could use for our study was to use the samples of hypertension-associated cardiovascular disease sub-study, and those participants were all free of diabetes at baseline. Then what we found was that, obviously, as expected, that the cholesterol levels and LDL cholesterol levels were reduced upon intervention. So this is a 12-month time frame we've studied here. But on top of that, there was also quite a substantial change in circulating microRNA levels in the statin group compared to the placebo group. When we then went on and went into cell cultures to study this further and did similarly an intervention with atorvastatin, and this was like a dose-escalating exercise, we saw that there were no changes at the cellular level in the primary hepatocytes, but when we looked at the medium, we saw that with greater concentration of atorvastatin, the release of microRNA 122 into circulation is reduced. And we know from previous studies that it's a bit difficult to see on the screen here, but so microRNA 122 does not localize to lipoproteins, so they cannot be found in there, but they're mainly found in exosomes. And it might well be that statins affect this release of exosomes into circulation and thereby lead to this decrease in concentration in the medium or in circulation. Okay, so finally, let's come to the longitudinal analysis that we wanted to conduct in that study. So, as discussed or as said before, so we focused here on the follow-up time between 1995 and 2010, so a 15-year risk or risk over 15-year time frame. So the Bruneck study is it's really quite comprehensive, and there's a close and good collaboration between the Medical University in Innsbruck and the local hospital there. Also, there's a huge advantage that the participants there are not very mobile, so very few. You have very little in and out migration of that catchment area, and that helps you to really trace people over time quite well. So we can really say that we have complete coverage for clinical events in all our participants, which is quite rare. And then also on top of that, so what we always did when we ascertained and diagnosed different diseases is when someone self-reported that disease, said, I have developed diabetes, we always confirmed this by reviewing medical records from GPs, but also at the Bruneck hospitals, as well as repeat laboratory measurements that we reviewed. So for the diagnosis of these different outcomes, so we looked at development of the metabolic syndrome of type 2 diabetes and cardiovascular disease. We used established diagnostic criteria, and you can see here on the right the number of incident events that had occurred over this 15-year time frame. And one thing to point out as well is that for the cardiovascular outcome, so we used a combined endpoint, combined of like myocardial infarction, stroke, or vascular death. And then for the analysis, so depending on which type we looked at, so we used Cox regression or a pooled logistic regression, which is a modification of the standard logistic regression that really makes use of the repeat measurements we had available in the study. And of course, we also, because we were interested in etiology and the first event that occurred in those people, we excluded people that had pre-existing diseases at baseline. So this is what our findings were like for the metabolic outcomes. So you can see here the outcome of metabolic syndrome and type 2 diabetes, and this is the odds ratio per 1 standard deviation higher level of log transformed microRNA 122. You can see those estimates on the right side of the null line. So there's a positive association, an increase in risk that was actually quite strong, especially for the metabolic syndrome. So you can see estimates here for the age and sex adjusted model, so 1.59, and for type 2 diabetes it was 1.39. When we then applied the multivariable model, so this model then further adjusted for socioeconomic status, smoking, physical activity, alcohol consumption, all well-known and established potential confounders in diabetes research, we saw very little change in these estimates. What we did see in a sensitivity analysis, just to mention that, is that there was some attenuation when we further adjusted for insulin resistance and for adiposity measures. But when we compared different subgroups, the association was robust, so including people at different ages, males and females having or receiving a statin or not receiving a statin, and a comparison of different clinical categories of the body mass index. The picture for our results for cardiovascular disease will look quite different. So you can see here that the overall result was null, so it was not significant with a p-value of 0.350. What we did, however, see is that there was a significant interaction even after adjustment for multiple testing, supporting a potentially stronger or an association in people aged 64, so younger. There was this discrepancy, of course, between the associations, clear associations for metabolic diseases and a null association for cardiovascular diseases, and we thought that this might also be explained by the natural history and of those different types of diseases, and it takes some time till changes and in the lipid profile then actually manifest into a cardiovascular event. So to summarize the findings in this particular study, so we used a multidimensional omics approach and combined epidemiological and experimental data. So our key findings from this piece of work is that we find a correlation of the liver-specific microRNA 122 with lipid species that contained saturated and monosaturated fatty acids that are produced by hepatic de novo lipogenesis. We found also a correlation with an adverse metabolic profile. We saw that statin intervention led to a reduced release of microRNA 122 from the liver, and we observed strong and independent associations of microRNA 122 with the long-term risk of metabolic syndrome and type 2 diabetes in a general population study. So this is what we found in our study. There are obviously, this was published in, I have to say, 2017 in diabetes, and there have been several developments in the area since then. Some things that I would like to just point out to you is that so there was one paper that looked at the correlation with proliferative diabetic retinopathy that found a link there. There are also several further studies that that observed that microRNA 122 is higher in type 2 diabetes patients that have non-alcoholic fatty liver disease than those without, and similarly higher levels in children with non-alcoholic fatty liver disease compared to healthy controls. If you would like to do some further reading and get a more broader overview of what has been published up to the year 2016, so that, I can also refer you to this paper that we have, a review that we have published in the European Heart Journal in 2016 that summarizes the putative role of microRNA 122 in all these processes. So as we discussed already, and furthermore, it also provides an overview of existing case control studies and different changes that we can see for metabolic outcomes, but also for cardiovascular outcomes. And most importantly, also to conclude, it is, of course, I presented to you the data on microRNA 122, because this was a hypothesis-driven approach, but there are, and I don't want to frighten you, there are many, many different microRNAs that are discussed in the literature, and this is a really excellent review that has just come out recently that discusses all the different functions that are proposed. However, in our study, we can only say that microRNA 122 in a population-based study was relevant for disease risk. So I just want to conclude by thanking my collaborators. So this was, I'm just presenting it here, and obviously there were many different people involved, from King's College London, the group that did most of the measurements for this study, the Medical University of Innsbruck, the Bruneck Hospital. We also collaborated with Enzo on this paper, Yale and UCL, and of course my funders, the Austrian Science Fund, and also the British Heart Foundation. So I look forward to discussing this now further. Thank you.
Thank you very much, Peter. Very interesting. Now, the talk is open for discussion, comments from Professor Dotta, Professor Triskita. It's coming. Microphone is coming.
Thank you for the beautiful presentation and for the results that you're bringing into the field. I have mainly two questions. The first one, you have clearly shown that MIR-122 is mainly located at the exosome level. So my question is, considering that when you measure circulating microRNAs, it's always a matter of techniques, approaches, and so on. My question is, do you suggest to measure these specific microRNAs isolating exosomes? You have a better yield, or is enough to measure in whole plasma? And so maybe you have a better performance of this type of measurement. The second question is, you have shown that the data in the mice where you, with the antagonist MIR-122, showing some kind of metabolic effects. You had probably shown in one of the reviews you showed at the end. Is there a specific signatures of genes that are modulated by this microRNA that can explain this metabolic effect at the liver level, for example?
Thank you very much. Yeah, so in terms of measurement, so as I've shown you, so these data are based on plasma and serum levels of microRNA 122, and we can only speculate if we had a measurement available directly in exosomes, that associations might be even stronger. So I'm not sure whether there's any data out there at the moment, surely not from a very big study, but I mean, it would be interesting to look into that. We didn't have these data available. And then the other question, so So in terms of the different targets and consequences that microRNA 122 inhibition had, so we saw these individual signals, and I think that in terms of the role in the different things that I showed, so in terms of packaging, of like the VLDL packaging and de novo lipogenesis and so on, there seems to be quite a nice match between changes at the gene expression level and proteomics level and what we see in the observational data. But also this is something in terms of the analysis, something that one could go into more detail. One thing that I thought and I would like to do, what would be quite interesting, is to look at genetic variants that are related to microRNA 122, but also the specific targets, and then use these genetic data to get a better understanding. But that is also something for a next step in this research. So.
Professor Trischitta, and then Dr. Bellia there.
Thank you for the nice presentation. If I got you well, I mean, you had two measurements, right? One in 2000 and the other one in 2005. Am I right?
Yes.
Okay,
exactly.
So the question is, when you try to predict type 2 diabetes or metabolic syndrome, have you had the chance to do that with both measurements? And if so, is the second one outperforming the first one, like anyone, I think, would hypothesize?
So in the main analysis that we did, we focused on the baseline measurement of microRNA 122, so in 1995. So it is true. So what we did is I showed you the pooled logistic regression, so this is a way where you can update different covariates in your model. We did that for the risk factors, but we didn't do that for microRNA 122 because we didn't have that for all the resurveys. It is plausible that could improve the prediction even a bit further, especially because there seems to be some variability in people, and then an accurate remeasurement of your exposure, so the level that the people are actually then exposed to at a certain time frame. So that might help, and this would be something to do, but we didn't do it in that paper. Yeah, thanks.
Okay, sorry. Alfonso Bellia, Rome. Thank you, Dr. Willeit, for your brilliant intervention. I was wondering whether the plasma level of this microRNA can be modulated in some ways by factors known to affect insulin sensitivity, such as the use of metformin, the level of physical activity, diet, and if you considered this point in your longitudinal analysis. Thank you.
Thank you. That's an excellent question. So as I've shown in the multivariable model that we did, we had to focus on specific variables and account for those potential confounders. But obviously, one could go beyond that, looking into treatment and looking into diet. But this is something we unfortunately couldn't do, also because of lack of power, even though it's a relatively big study for a population-based study, but there are some limitations of what we could do, yeah.
Miami. Thank you for sharing this nice work. I had two quick questions. You showed a correlation between plasma and serum measurements of MIR-122 at two different time points. And in the earlier time point, 1995, as compared to 2000, the correlation was much less. I think you mentioned something like that the levels had changed in the patients, but could it be that the sample had changed, that there was degradation in one of the two at least, and you lost correlation? And because you have shown in your design that you had, I think, about 5 longitudinal samples, I wasn't sure that you've measured all of them, all the time points, and if you've analyzed the data longitudinally to see whether, for example, there are changes in MIR-122 level over time, and is the rate of change or the magnitude of the change that could actually predict disease in the future.
Yeah, this is excellent. Yeah, it's a very good comment. Of course, so there can be, or there could be, some degradation over time, and the levels could be related to storage time, so which was different to those at those two time points. It is true. So the measurement of microRNAs is in general a bit of a tricky field. So this is also why we kind of like collaborated with some of the leading groups for that research, and there are many things that need to be taken into account. We only had the measurements in 1995 and 2000, so we can only look at the correlation between those 2 time points. It would be interesting to see how dependent on the time point, whether you get the reduction in the absolute levels that we measure. Yeah.
Other question? Otherwise, okay. First, Lorenzo.
Nice presentation. I have a doubt. Your microRNA correlates or do something on lipid synthesis and oxidation. So, because in the liver, fatty acids go one way or the other, which is a primary regulation, synthesis or oxidation?
I'm not sure. Yeah. So this, I think, is also something that where the data is not absolutely clear. I'm afraid. Yeah.
I have a couple of questions. One is, it's clear that the main effect of this microRNA seems to favor the accumulation of fat where the fat is not well accepted, like liver, or, for instance, also is associated with the increased incidence of metabolic syndrome. And usually metabolic syndrome is driven mostly by the accumulation of fat in the visceral tissues. So I'm wondering whether you or others have measured the MIRNA in correlation with the accumulation of fat in skeletal muscle or heart or in epicardial fat, for instance, just to see a general phenomenon of accumulation of fat in other sites that are not the adipose tissues. And the second question related to this, indeed we have one insulin synthesizer, which is 5-glitazone, which is exactly able to remove the fat from these tissues. And I wonder whether there is some data showing that there is a modulation by PPR gamma agonist.
Yeah, this is interesting. Yes, so this is actually, so in some part in terms of adiposity, we looked at like the traditional markers, body mass index and waist-hip ratio, but this is something where we could go into further detail, and there are different other things we've assessed in the Bruneck study. So this is, I think, an aspect where we could deliver some additional results and provide some further insight. Obviously, so in terms of therapeutic intervention, it's an observational study where only few or a small proportion are diabetic, and so therefore the proportion of people that receive certain therapeutic interventions is usually quite small. So I think a clinical trial setting would be better to study that further.
Peter,
yes.
From the mechanistic studies, it seems that there are more relation between lipids and biochemistry of lipid metabolism, molecular biology of lipid metabolism, as compared to the biochemistry and molecular biology of glucose metabolism with this kind of pill now.
Yes, I would agree with
the data, longitudinal data show a good association between the incidence of diabetes. So there are some pieces of the puzzle that needs to be identified and put in the right place. Do you think?
Yeah, so this is also something that we found quite fascinating. So having this main focus on lipid metabolism, but then finding an longitudinal association with metabolic disease, but not cardiovascular disease. And so this is, so we have to better understand how it is really related to de novo lipogenesis, how it's related to non-alcoholic fatty liver disease, and what the potential links are. So one, for example, could be affamine, that is relevant as well for the development of diabetes, but also the zinc alpha 2 glycoprotein, which I showed briefly, where which is important, as I mentioned, in the insulin sensitivity of peripheral cells. So these are potential links where somehow we can see how those might be biologically linked to each other and could explain the effects and observations we see in the Bruneck study.
One more question. The last one, please.
You tested other statins other than atorvastatin, maybe rosuvastatin?
I'd love to, but we didn't. We had data from the ASCOT trial, but there's actually, so I'm coordinating a consortium for statin trials in Innsbruck. It was more focused on lipoprotein A, but this could be a source maybe that one could look at different types of statins and as well as different dosages in the future.